So the second major biochemical pathway that we're going to investigate and study is photosynthesis. Now, again, when we look at kind of the energy flow through an ecosystem, we know that light is coming in to the system and being absorbed by what's known as an autotroph, in this case, a green plant, that can take that light energy and convert it into a biologically usable form, organic molecules in O2. These are the things that you take and all eukaryotes take basically in one form or another and burn in cellular respiration in their mitochondria to produce CO2 and water, which again are the raw materials that in the presence of chloroplasts and light energy we can convert back into sugar. Ultimately, we make ATP for the cell to use, and that is our biological kind of energy currency. Now, when we consider this from an organismal point of view, we talk about autotrophs. Autotrophs being organisms that can produce their own food, the vast majority of which are going to be green plants, photoautotrophs, using sunlight energy, fixing CO2 to make food. Heterotrophs are what we are. We're other eaters, other, trophs, eater. And you can break that into many categories of herbivores, they eat plants only, carnivores, they eat other animals, or omnivores, which eat plants and animals. We'll discuss this further in 213 and other courses. Now, I want to orient you over the next couple slides to where photosynthesis is occurring in green plants. So we're going to look at a little bit about plant anatomy, and we know that leaves are the major location of photosynthesis. Those stems will photosynthesize as well. It's the green color from the pigment chlorophyll, and we'll look at that more closely as we go. Light energy is going to be absorbed by the chlorophyll, and that's going to drive ultimately the synthesis of organic molecules in the chloroplast. CO2 is going to be our raw material. That's our carbon source, right? So CO2, the inorganic carbon in the atmosphere, is actually going to be built into sugars, organic molecules. And the byproduct or the waste of that is oxygen, which is going to exit the leaf. So we're moving gases in and out, so we have what's called gas exchange through what are called stomata. So singular stoma are seen generally on the underside of the leaf. This is a slice of leaf anatomy. And you can see you have the upper epidermis, followed by the palisade chlorophyll, or oftentimes known as the palisade parenchyma, and then the spongy mesophyll. But the anatomy of the leaf is set up to maximize gradients to allow CO2 to keep coming into the leaf, to allow O2 to keep coming out of the leaf, and to funnel light energy down from the top of the leaf into the interior of the leaf. So what we're going to come across later is this idea that because water vapor is also exiting the leaf with the waste product of oxygen, and we need to have ways to get CO2 into the leaf that also allows water vapor to leave, that can become a resource problem if water is limiting. So if we keep losing water and we can reduce water loss by closing our stomata, we also cut off our CO2 supply. But we'll talk about that later. I just want to plant that idea in your head now. So again, some of the vocabulary: palisade parenchyma, the long columnar cells, the top of the leaf, the spongy mesophyll. This is where a lot of most of the photosynthesis is occurring. Now, in these cells, we have chloroplasts. So we know about chloroplasts as an organelle. 30 to 40 chloroplasts is typical. And inside the chloroplast, we have the thylakoids. Remember, it's that third membrane, outer envelope, inner envelope, thylakoid membrane, which is stacked in what are called grana. And these little stacks are built in a way to maximize their light absorption, and it floats in what's called the stroma, the fluid inside the chloroplast, in which the chloroplasts are kept. So again, this slide allows you to kind of dive in from the leaf, zoom in, you see that cross-section of the leaf, there's the stomata, CO2 in, oxygen out. Then we zoom in on a single leaf cell, and then zoom again into the chloroplast. And I like this slide particularly because it shows the chloroplast with an outer envelope and an inner envelope being relatively clear, and then the green part of the chloroplast is actually localized to the thylakoid, and that is in fact very accurate. So the chlorophyll, that is the green component, is in fact only in the thylakoid membranes. Now, if we look at a summary of photosynthesis, we can kind of see what we've talked about already, that we have CO2 coming in, it's our carbon source, we have a utilization of water and light energy, and out the other side we get carbohydrate, oxygen, and water as a byproduct. So what we're ultimately doing is oxidizing water. which is really hard to do because we know oxygen is such a good electron acceptor, right? Like it's very electronegative. But with the power of sunlight, we can actually create an oxidizing agent that will actually oxidize water. And then CO2 is going to be reduced. So we're adding electrons to CO2 and building up molecules. So we think about oxidation as ripping those electrons away, and reduction as building them up by adding electrons. All right, so if we look at that, we can see the reactants and then the products. And it's not necessarily very straightforward when we just look at the summary, but what we know then is that this summary reaction is actually the summation of many reactions. And we can categorize those reactions into two major groups: the light reactions and the light independent reactions, which are also known as the dark reactions or the Calvin cycle. Now, the next slide, this slide, we're going to see this all again and again, because this is going to be kind of our summary slide. It is the cross-section of the chloroplast. We can see the light reactions on the left, and then the Calvin cycle, or the light independent reactions, on the right, which is actually the point at which we're going to fix CO2. All right, so let's talk about the light reactions. The light reactions summary are going to split water, release oxygen. We're going to produce NADPH. So it's very similar to NADH that we looked at when we looked at cellular respiration, and it serves the same function as an electron carrier. It gives us the ability, it's reducing power, it gives us the ability to add electrons to another molecule and build it up. So the light reactions are going to generate ATP from ADP by what's called photophosphorylation. So in cellular respiration, we talked about oxidative phosphorylation. Here, we're going to use photons of light to power a system that will allow us to use an ATP synthase, just like we did before, with a proton gradient, the proton motive force, to make ADP into ATP. Now, the dark reactions, then, are the light-independent reactions. And we want to focus on them because they are going to be the point where we attach carbon molecules together to start to make sugars. They're going to use the ATP and the NADPH from the light reactions to fix carbon. So this is how we're going to start to build organic molecules. Now, photosynthesis is very much associated with the green chlorophyll pigments. So let's focus a little bit on the pigments and look at kind of where the rubber hits the road for absorbing light energy. So pigments are substances that absorb visible light. And if you go on into more physical chemistry, you can study the structure of pigments and how they absorb light, but we're not going to do that here. But we do know that different pigments absorb different wavelengths based on their interaction with the wavelength of light that they absorb, right? So not all wavelengths are going to be absorbed by a single pigment. And leaves appear green because chlorophyll, the major pigment for light absorption, doesn't absorb green light energy, right? Light of green wavelength. So we can look at what's called an absorption spectra when we analyze these different pigments, and we can see that there are two major peaks in the low blue range and in the high red range. And in the middle, where it's green, we have very, very low absorption. All right. So that is why, plants look green. Now, there are accessory pigments that we'll talk about later, the carotenoids, and there's a variety of other families of accessory pigments. But these are going to extend the range to which a leaf, an organism that does photosynthesis, can absorb different wavelengths of light. Right? Now, if we look at the chlorophyll molecule itself, we can see that there's a prosthetic group of magnesium that is bound, and this porphin ring is what is the head of the molecule, and it's bound. It's holding that magnesium. It does have a hydrocarbon tail. This is like it's like a lipid, right? So it's an integral part of the membrane, which I'll show you pictures. Now, there are two major types of chlorophyll that we're going to see in land plants, and those are chlorophyll A and B. And you can see the difference between them, and this is just the difference between that methyl group and that CHO. Right? And that small difference changes kind of the resonance of the entire structure to shift the absorption of those two pigments. So if you go back to this and look at the absorption spectra at the top there, you can see the slight difference, the shift in chlorophyll A versus chlorophyll B. And that, again, comes down to just a handful of atoms in this very large molecule. Right? So chlorophyll A is the main photosynthetic pigment, and chlorophyll B and the carotenoids are generally considered accessory pigments. Now, what we really do then is when we collect light, we create arrays of pigment molecules called antenna complexes. And really what we're going to do then is kind of collect them all almost like a parabolic mirror and shuttle them to the center where we have what's called the reaction center, where there's what's called the special pair. And we're going to talk about that more as we talk about the details of the light reactions. But ultimately I wanted you to get a feel for what this looks like in the membrane, where you have all these pigment molecules now surrounding this protein complex, and they're ultimately shuttling the energy of the photons that they're interacting with into that complex, and that's going to allow them to oxidize water. So we'll talk about the details of this complex and how the photosystems work in the light reactions recording.